Shalom. Good morning to everybody. Thank you for the introduction. I also would like to start by thanking the organizers for inviting me to this 3 or actually 4-day visit to Israel. This third day is the day for public lectures. Yossi Loyan told you that the person who really has been the driving force behind this event is Professor Tamar Dayan. And I know that this is true because she actually came to my office in Helsinki, Finland, personally to invite me to participate in this meeting. I guess she had some other business in Finland as well, but I thought it was very nice. I would like to start by telling a small anecdote about Professor E.O. Wilson, whom many of you know by name, and who we could consider to be the grandfather of biodiversity and biodiversity research. Professor Wilson is professor in the Harvard University in the US. 1990, the Harvard University magazine sent a message to 8 famous professors in the Harvard University and asked them to name the threat that these professors would consider to be the most severe threat to humankind in the coming decades. The answers included threats such as uncontrolled growth of mega cities, infectious diseases, nuclear war, overpopulation in general. Even capitalism was mentioned as the most severe threat. Professor Wilson's response was the loss of biodiversity, and he justified this reply by observing that humankind can actually recover from all these other threats, even nuclear war, even capitalism, in a matter of tens of years or at least in hundreds of years. But we can't recover from the loss of biodiversity in hundreds of years, not in thousands of years, not in tens of thousands of years, not even maybe in millions of years. It most likely takes tens of millions, or at least a 10 million year period before, in the subsequent course of evolution, the lost biodiversity has been recovered. And we know this because following the previous mass extinction on Earth, it has taken millions of years, up to 10 million years, for biodiversity to recover. Well, the reason why Professor E.O. Wilson and many others are concerned about the loss of biodiversity is that it is so fast at the present time. My 3 figures in the title, they refer to the past rate of loss of biodiversity on Earth, the rate that Dan Simpeloff called the background rate, which is the rate of extinctions which has occurred during the past 500-600 million years most of the time. And we know this rate based on fossil record. Now, oftentimes the rate is expressed as the numbers of extinctions per 1 million year. But because it's very difficult for me, and I'm sure also for you, to really understand what means 1 million years, I have converted the rate to percentage extinction in 100 years. We can all understand what is percentage, and we can understand what is 100 years. So the background rate of extinctions, the rate of extinctions which prevailed on Earth before the human influence, has been estimated to be 0.001% in 100 years. So that fraction of species has been estimated to go extinct in 100 years. So not much, and so little that actually the rate of evolution has been fast enough to compensate for those extinctions. But the present rate of extinctions has been much accelerated. We heard from Stuart Beeman. I think we will hear more from him in the afternoon about the current rate of extinctions on Earth, which is very roughly now set at 1% in 100 years. So 1% of all species of animals and plants are going extinct in 100 years with the current rate. Now that doesn't yet sound very alarming. Unfortunately, the rate is not perhaps even now while I'm speaking anymore 1%, but in any case we know that it's accelerating, and researchers now consider that the rate is more than 10% in 100 years by 250, maybe much more than 10%, maybe 20, 30%, or some people think it's even greater. Now that sounds such a high figure that you might consider whether this is even a realistic estimate. In the lower part of this slide I have indicated the numbers of red-listed vertebrate species, birds, mammals, fishes, amphibians. At the current time we know well how many vertebrates there are. We know pretty well what is their level of threat, and roughly 20% of all vertebrates are currently considered to be threatened, which by definition means that they have a significant risk of extinction within a period of 50, 100 years. Now the main reason, as we have discussed over the past 2 days, for the rapid rate of extinctions, both locally, nationally and globally, has been and still continues to be habitat loss. This figure shows the global threats to biodiversity in mammals, amphibians and birds, for which the data are best, and as you can see, habitat loss is the main culprit. Now this figure is some years old. Climate change is not mentioned here. At present time, and particularly in the future, climate change is going to be yet another major threat. So those were my 3 figures, and particularly the middle one, 1% in 100 years, is very easy to remember. Now, given that habitat loss really is the main cause of species extinctions, I would like to describe with a couple of examples 2 key concepts and phenomena which relate to the way that habitat loss is threatening the viability of species. And these 2 concepts are extinction threshold and extinction depth. My first example comes from my own long-term, well, when I say my own, that means, of course, my research group, students and associates. Our long-term research on a species of butterfly, the Glanville fritillary, which you actually have also in northern Israel. We have studied this species for the past 20 years as a kind of ecological model system in northern Europe on a large island where we have mapped, and we now here means a huge number of undergraduate students, have mapped the appropriate habitat, dry meadows for the butterfly. There's a large network, about 4,000 such meadows on the island. On this map I have colored with different colors sub-networks, different parts of this large 4,000 meadow network, which sub-networks are, from the point of view of the butterfly, are dynamically more or less independent because the distances that the butterfly can fly are only a couple of kilometers. So if there's a butterfly population, or what I call a meta-population, living in such a sub-network, that is more or less independent of the other such meta-populations. And as you can see, the colored sub-networks, they differ greatly in terms of how many meadows there are in each one of them. You can't see that there's also variation in the areas, there's variation in the average distances to other meadows. Now, on this graph I have plotted on the vertical axis a measure of network structure which measures how favorable the network is from the point of view of the ecology of the butterfly. If you are here at the right-hand end, the network is really good. It has many meadows. Those meadows are on average large, which is good for the local populations. They are not much isolated, which is good for dispersal and gene flow. If we are at this end, the situation is the opposite. The subnetwork is small, in other words, has only a small number of meadows. The meadows themselves may be small and they may be isolated. This measure here, it combines all these factors into one single real number. And on the vertical axis, we have the empirically measured abundance, commonness of the butterfly in the different subnetworks. So each dot here is for one subnetwork. If you are down here, the butterfly is completely absent from the subnetwork. If you are up there, the butterfly actually occupies all the meadows within the subnetwork, all the habitat which is available in the fragmented landscape. And the main point here is that when the quality of the network becomes more favorable for the butterfly, we go towards right, the butterfly becomes more abundant, as you can see, but the response of the butterfly is not linear. It's definitely non-linear, and there is a point along this axis which gives the measure of network quality, where suddenly the butterfly actually occurs in the network. If the quality is below this limit, the butterfly in this empirical example is absent from all the networks. If we are above, it is present. And this limit we call the extinction threshold. And the message here is that the fragmented landscape needs to have a certain minimum amount of habitat for the species to have viable population. And it's important to understand that this response really is non-linear rather than linear. I take another example from another part of the world, from the coastal forest in Brazil, which is one of the biodiversity hotspots on Earth. Many species, many endemic species. These results are from the work of Renata Bardini in Sao Paulo University. She and her students, they have been studying the occurrence of forest-living small mammal species in different landscapes which are more fragmented or less fragmented. Landscapes where either 10% of the forest cover remains, 30% of the forest cover remains, 50% of the forest cover remains. So these are the study areas. Nearby there is a more continuous forest left. And in these results we observe exactly the same phenomenon. Each line here is for one forest specialist small mammal species. Here is the forest cover from 100% forest cover to 10%, which was the minimum. Here we have a measure, again, of the abundance of the small mammal species, basically telling us if in a fragmented forest landscape there are, let's say, 20 forest fragments, which fraction of those fragments were occupied by the focal species. You can see that there are common species which occupy most of the fragments in the fragmented landscape. There are rare species, but in both cases there appears to be a sudden drop in the commonness of the species from the sort of normal level, whether the species is uncommon or common, to nothing basically, between 30% and 10% forest cover. So again, the response is non-linear, and in this case the extinction threshold for this forest specialist small mammals is somewhere between 10% and 30% forest cover. So that was the concept and phenomenon of extinction threshold, and I then move on to the other key concept, which is extinction depth. And I do this with another forest example, this time from North America. A botanist called John Curtis did some real pioneering, important work in Wisconsin in US in 1950s, a long time ago. He surveyed a large number of forest stands, all the black dots here in Wisconsin. Another botanist, Dave Rogers, a few years ago got the great idea of going back to the notes of John Curtis. John Curtis had kept very careful notes exactly where he had sampled the plants and which plant species he had discovered. And Dave Rogers went into exactly the same forest stands if they still existed, some didn't, and he did a comparable survey. And Dave Rogers additionally measured the cover of forest around each forest stand within a radius of 5 kilometers. Now what I have done here is to plot for John Curtis's original data the number of vascular plants which are forest specialist species in the different forest stands, so each dot for one stand, against the percentage forest cover within 5 kilometers. And what we can see is that there is no relationship whatsoever, which is kind of surprising because, as somebody mentioned yesterday or the day before yesterday, typically what we see in ecology is that more you have habitat for focal species, more you tend to have species in the community, the species-area relationship. But it's not here. However, in Dave Rogers' results, when he studied exactly the same forest stands, there is a definite species-area relationship. So in these stands, which had little forest around them within 5 kilometers, the number of vascular plant species was definitely smaller on average than in the stands which were surrounded by lots of forest. Now you might think that this might be due to the fact perhaps that forest cover changed between 1950 and 2005 and species just responded to that change. Well, actually that is not the case because as a matter of fact the forest cover in this area has remained about the same since 1950 and actually before 1950. Here is one example from one forest stand. Some people will recognize this as a famous figure from the book of MacArthur and Wilson illustrating forest fragmentation in this area in Wisconsin. I used Google Earth that Stuart Pym yesterday publicized, indeed very practical tool. It was easy to locate this very area and it was easy to map the current situation with forest, and as you can see, no much change. No much change since early 1900. So it's not really, to me, plausible that the striking difference between 1950 and 2005 in the results would be due to some environmental change, anyway, a change which you could observe with Google Earth between these two periods. However, as we can see, there certainly has been lots of forest loss and fragmentation in earlier times. And what we are proposing with Dave Rogers and David Mladenov is that the difference that we see here is due to a drop of the species number in the forest stands, which both in 1950 and 2005 are surrounded by little forest. And this drop has been due to a time-delayed response of species due to a previous loss and fragmentation of forest. So as you saw from the example, before 1950, long before 1950, there was forest loss and fragmentation, and we are proposing that the species here, they have declined in these stands between 1950 and 2005 due to that old loss and fragmentation of forest. But it's only that it has taken a long time for the species actually to decline, and it's easy to understand why, simply because the populations don't demographically respond instantaneously to a change in the surrounding forest. So we call this phenomenon extinction depth, a decline and extinction of species due to past loss and fragmentation of forest. And very roughly, if you take these figures at their face value, there was a 50% extinction depth in these stands which are surrounded by little forest in 1950. So these are the two key concepts and phenomena that I would like to emphasize in this context. Extinction threshold, species responses to habitat loss and fragmentation are non-linear, therefore you should expect unexpected changes if and when habitat is being lost. And secondly, extinction threshold, it is important to understand that there is this time-delayed response, which oftentimes means that we are likely to underestimate the level of threat to biodiversity because of extinction depth, simply because we don't perhaps realize that the species are not at equilibrium now, they are still slowly responding to past changes in the environment, which means that even though currently we don't cause any further loss or fragmentation of habitat, many species may still be declining because it takes such a long time for them to decline. So, my two concepts, and then the hypothesis. So it's a fact that there's this what we might call a global megatrend in biodiversity. Biodiversity is declining locally, nationally, globally. Here are three indices. LPI is the WWF Living Planet Index, which summarizes information for thousands of populations and species worldwide, and based on that index, biodiversity has been lost at a real fantastic rate. About 30% of the index value has been lost in only 30-40 years. So, a global megatrend. Now, clearly there are also other global megatrends, and this graph here shows another dramatic change in something, and that something is the incidence of asthma, hay fever, in this example, but also other allergic diseases, also other diseases which have an inflammatory basis. In this case, in a Finnish population, but I would imagine that also in Israel you have a similar kind of increase in, let's say, allergies. Now, 2.5 years ago, I just happened to start talking with two medical doctors, Leena von Hertzen and Tari Hahtela. We talked yesterday afternoon how important it is for people with different disciplinary backgrounds to have an opportunity to interact on a personal basis without anybody saying that you should interact, but you just happen to interact. And this is, I think, a very good example of such an interaction. We started thinking about these two megatrends, the declining biodiversity and the equally dramatically increasing incidence of allergies and other inflammatory diseases, which, by the way, include things like type 1 diabetes, some cancers belong to this category. And we came up with the hypothesis, not entirely original, but nonetheless, which was to us an exciting idea, that perhaps we are here seeing the interaction between three complex systems, one of which is the environmental biodiversity, including environmental microbiome, all the bacterial, fungal species in the environment. That's one complex system. The second complex system is the microbial system that we have in and on our own bodies. As you may know, my body has actually 10 times more bacterial cells than my own cells. In the gut, on the skin, in the respiratory tract. A hugely complex system with thousands of species. And the third complex system is our immune system. It doesn't have many species, but does have many different kinds of molecules, maybe not thousands of molecules, but certainly hundreds of molecules. And the hypothesis is that these three systems are interacting in the way that the environmental microbiota has some influence on the composition of our commensal microbiota, which, as is now well known in immunology, is interacting with our immune system, stimulating various types of cells in the immune system, and oftentimes with the consequences of enhancing our immunological tolerance. The hypothesis is that if this connection is somehow perturbed, one example might be you taking antibiotics will be messing up your commensal microbiota, but another sort of perturbation might be microbial deprivation in the sense that you and your commensal microbiota is interacting less with the environmental microbiota than you used to do. That might lead to this immunological tolerance being broken. Now, this idea is not new in the sense that, as I'm sure many of you know, there's this general notion that if children are growing up under too hygienic conditions, this is not good for the development of the immunological tolerance for the reasons which I just explained. Lots of empirical case studies showing that, for instance, growing up on a traditional farm in Europe is particularly healthy thing. So what we are really doing here is kind of taking the hygiene hypothesis out of doors, and we are proposing that the broader environment is also important in this context. Now, what I would like to present here is a little case study which we have just completed to test this general notion. And the case study involves a random sample of 118 school children living in eastern Finland in a landscape which is heterogeneous. There is one little town, villages, and then rural area, children living with their families in isolated farmhouses. So this is Finland, this is Russia in eastern Finland. And really what we wanted to think about and empirically examine is the possible interactions between these four pieces: environmental biodiversity, our commensal microbiota, and we use the skin microbiota, which is convenient to sample as a representative sample of that, atopic sensitization, or what you might call allergic disposition, which measured, as I will show on the next slide, and then we did also something about the immunological response in these individuals. So the allergic disposition or atopic sensitization was measured from the blood samples by measuring the level of immunoglobulin E antibodies for a set of inhalant allergens, which is something that medical doctors who are working on allergy do in this context. And quite conveniently in this sample, the distribution of these antibody levels was bimodal. So we denote this first mode here as the healthy individuals, low level of antibodies in their blood, and these are the atopic individuals. Well, then we went back to Google Earth and measured the environmental biodiversity in terms of land use types in the surrounding of the homes of the individuals within a radius of 3 kilometers in this context. Now these people are living in eastern Finland, so there's quite a bit of forest around their homes, mostly highly managed forest, but forest nonetheless. There's agricultural land, built areas, water bodies, and so on. We took these skin bacterial samples, and using the current metagenomics methods, we identified, using the so-called barcode technique, the composition, the structure of the skin microbiota in these samples. Very diverse. Altogether, 572 bacterial genera were identified in the pooled sample. And who knows how many species? It's not really possible to tell the bacterial species, however you define them with these kinds of methods. This slide shows the main bacterial classes in the sample: Actinobacteria, Bacilli, Clostridia, beta, alpha, and gamma proteobacteria were the dominant groups. Now, the first result, or the first analysis that we did was to examine the relationship between the land use types in the surroundings of the homes and then the composition of the skin microbiota. And I'm now here summarizing the variation both in the land use types and in the composition of the skin microbiota using a multivariate statistical technique which Mark Feldman spoke about yesterday. This is one of the simplest multivariate statistical analyses called principal component analysis, which just summarizes the common elements in the variation in the land use types and in the bacterial composition. What the result shows that indeed there is a significant relationship between the land use types in the surroundings of the homes and the composition of the skin microbiota. And by and large, it goes in such a manner that if there's lots of forest and agricultural land in the surrounding of the home, there are lots of proteobacteria on your skin in this population. If there are built areas and water bodies in the surrounding, you have other bacterial classes. There is, of course, there's a huge amount of variation, but there's also a clear trend there. So land use types appear to affect skin microbiota. There's also a highly significant relationship between the land use types in the surrounding of the home and the atopy, whether the individual was healthy or allergic, if you want. Now, these are a bit low-quality graphs, but you can see here that on the vertical axis, on the horizontal axis, I have again this principal component, which is summarizing the land use types. We have forest here, agricultural land, at this end built areas and water bodies, same for here. And you can see that in the case of the healthy individuals, they are mostly living in areas where there is lots of these vegetated habitats in the surrounding of their home. In the case of atopic individuals, their surrounding has more built areas and water bodies, and this is a highly significant difference. So land use types definitely influence atopy. Next step was to ask about this skin microbiota, and to my very great surprise, we got one striking, what I would consider to be very striking result, which is shown here. In one class of bacteria, gamma proteobacteria, the generic diversity, how many genera of gamma proteobacteria you have on your skin, and we have here kind of scale that by the total number of bacterial genera in the sample, which is important because the sample size varies. We see this very, very striking difference, such that healthy individuals, they have many genera, high generic diversity of gamma proteobacteria on their skin in comparison with the atopic individuals, black dots, which have a lower diversity of gamma proteobacteria on their skin. Now, some of you might ask whether the causality here truly is from the composition of the skin microbiota to atopy. Might it not be possible that if you are an allergic person, atopic, that might influence the composition of your skin microbiota? Now, that is a very good question. We have a number of lines of evidence, however, suggesting that the causality is going from here to here, and I will show you the strongest piece of evidence. Now, further collaboration with a further set of researchers with different expertises, in this case immunologists. We recruited a couple of immunologists who measured the expression of various what are called cytokines. These are kind of small signaling molecules which are important in the immune system in blood samples taken from these individuals. And particularly, I'm here focusing on something called interleukin-10, which is one of the most important anti-inflammatory cytokines in humans. So this is a molecule which is suppressing inflammatory response, and thereby is preventing allergy by kind of making the body not to react in an inappropriate manner to allergens. So that your body doesn't care about almond pollen, which is a nonsensical response because it's not harmful. If you're allergic, your body is responding to such almond pollens or some other pollens or some other allergens. Now this interleukin-10, an anti-inflammatory molecule, is sort of suppressing that kind of inappropriate response. And what we found was yet another most striking result. And at this time, we actually went down to the level of genus. This Acinetobacter is a genus of gram-negative gamma-proteobacteria, so in the same class of bacteria than the other associations. And we found that in the case of healthy individuals, these are the open circles are the healthy individuals, there is a highly significant and strong relationship between the abundance of Acinetobacter on your skin and the expression at the RNA level of this interleukin-10 cytokine in your blood. And what it suggests is that these bacteria are stimulating some cells in our immune system, produce this anti-inflammatory molecule, which is suppressing inflammation. In atopic individuals, as you can see, the black dots, the relationship is completely different, and the interpretation is that now this immunological tolerance has become broken. Now, a most striking result is that this analysis identified this particular genus, Acinetobacter, our skin, out of 572 bacterial genera. And this is really exciting because other research done on the mouse model has actually used this very same bacterial genus and has shown experimentally that yes, in the mouse model, Acinetobacter suppresses asthma and allergic reactions. So another very strong piece of evidence suggesting that the causality really is from the composition of the skin microbiota to atopic or allergy. So I'm running out of time. I just draw two conclusions here. What I was able to present was a subset of the results that we have obtained in this study. The full results you can read in a paper which is coming out very soon. And what the results really suggest to me is that if you take the results at their face value, they suggest that more contact we have with environmental biodiversity, including the microbial component of that, that means something which of course we don't really understand that well yet, something to the composition of our own commensal microbiota which has beneficial effects on our immunological tolerance. And that means that indeed these two megatrends, fast declining biodiversity, rapidly increasing incidence of inflammatory diseases, they might have something to do with each other. Now, I'm an ecologist, not an immunologist, and so I obviously have a pretty naive perspective on these things. However, I'm not so naive that I would claim that this explains completely what we see in the increase in the civilization disease in our societies. But what I'm suggesting is that we are here perhaps seeing a contributing factor. Our reduced contact with biodiversity may be a contributing factor towards the rapidly increasing prevalence of allergies and other chronic inflammatory diseases in developed countries and particularly among urban populations. Thank you very much.